Ready? All right. Let's get over here. I didn't even get to the topic for today. Shows you how inefficient I have been. Kind of got not quite to there. So I thought we'd finish today, but in chapter 1, which is introductory, but didn't. I did sort of a stream of consciousness lecture. And I got back to this thing. Nevertheless, I always thought the theme was going to be something called homeostasis, and we'll touch on it. But really, we'll finish it up on Friday, and it's probably adequate time for the first chapter. Homeostasis is the state of internal balance that's just magnificent, and it really keeps everything, no matter what the conditions are, inside it. We'll talk about negative and positive feedback, but I don't want to give that short shift. I have a lot of other sort of material to cover about living things, and that's kind of what you were seeing up there. I apologize for my handwriting, but you're stuck with it. When you're a doctor and wrote prescriptions for 25 years, it's just, we have a course, how to write illegibly, which is why I mostly type. That's when you'll see notes that I type them out for you. It's easier. So all that aside, that's kind of where we left off. We were doing that initial construct that's there. Homeostasis, the beauty of that is effectively, no matter what our external environment is. Chilly this morning, I walked the dog, sweater on. Maybe, last week I was about the same time. So 7 o'clock, I got a pair of shorts and t-shirt. We can adjust to that external environment. We're one of the organisms that's fortunate enough to do that, for a lot of other organisms. But even at the same time, we're able to maintain other organisms, animals in the wild. I mean, we live in an area where it's deer, rabbits, squirrels everywhere. It's just tons of stuff, which is a lot of fun. But I mean, how do they survive with all this? Again, their internal environment is kept relatively well insulated, so to speak, is able to deal with it by changes that's made inside, and we're going to address those. Perhaps not today. So effectively, if we're going to be any living thing, and particularly we're talking about, in this case, we're dedicated more to ourselves, and particularly to animal type cells that are there. We have eukaryotic cells. That's EUKARYOTIC. All it means is, it means with a kernel or with a core. It's a cell, and there was the nucleus, but it isn't just the nucleus. Key issue is membrane-bound organelles. The idea of membrane, which we'll touch a little bit on today, and really do a lot on, we're talking about movement of protein, has a tremendous amount to do about things within our cells being able to work independently. It's a level of complexity. It doesn't mean it can't be impactful. Bacteria don't have membrane-bound organelles. They're a single-cell entity. Yet they affect us dramatically, frequently both adversely and positively. A misconception about them. They're bad guys. Listen. So, cells, membrane-bound organelles, we are, by definition, made out of cells. That's what cell theory is. So all living things are made out of cells, and all cells come from pre-existing cells. Basically, that's cell theory 101. By definition, we have to have within those cells both DNA and RNA. Down the road, you'll see DNA has a couple of significant roles. One, basically, the genetic code directs the proteins that are made and effectively creates what we are, so to speak. At the same time, DNA has to replicate in order for the cells to reproductive activities. Reproductive activities, as you will see, in our more complicated cells are both asexual and sexual. We do, comparatively speaking, if you think over a lifetime, very little in the way of sexual reproduction on a cellular basis. We just don't. Even though people think there's a constancy to the process, it isn't as great as you would think compared to the asexual nature. And you'll hear me say it again and again, so we might as well do that reflection. We don't repair ourselves, we replace ourselves. We replace ourselves by making new cells. You heal from something. If you can't replace it, you're going to have scar. You get into an area that does not have the capability of replacing itself, it heals with scar tissue, fibrous tissue, whatever you want to describe it. There. So DNA's job, so to speak, to get back to that concept, DNA has to provide the framework for both kinds of replication, a little more specialized, obviously, sexual. We don't even get to that to the very end of it. And RNA, which has several roles, including creating a platform to make proteins and basically developing the construct and the assembly of said proteins. You get to all of that. But that's where DNA and RNA come in. Now, that gets back to the membranes. The whole idea, we'll touch on it a little bit today, is the concept of semi-permeability. All living membranes have semi-permeability. I'm sure it's not foreign to many of you when I talk about something called the fluid mosaic model. Fluid mosaic model is what a living membrane is. Okay, and we'll get to it in a more elaborate manner. This is also introductory. Fluidity means it can change shape, it can open, it can close. I mentioned it in passing yesterday. There's a distinction. We were talking about macromolecules, proteins, and DNA and RNA, and complex carbohydrates. The oddball is fats. Membranes are made of fats. Fats aren't bonded together. They just kind of come together. Because they come together apart and go back together at various signals. That's how membranes open and close. How we ingest or bring food in on a cellular level is the membrane opens up. If anything, the membrane then surrounds the food, pulls it in, embraces it on food. I think it takes it all in. And how do we excrete things? We do the reverse of it. We spit it out. That's opening and closing of membranes. That's fluidity. The semi-permeability, the idea comes from proteins that are embedded. That's the mosaic. They're different. Mosaic. They move around, mosaic. They can open and close as signals. Sometimes they're always open. And we're going to go over all these different membrane proteins when we get to the cells. Again, this is all intricate set framework, contextual. And they might always be open. We call them pores. So our cells generally are porous. Not every one of them. You're going to see some that are impermeable to water. They're very specialized. They create specific boundaries. The most famous of those is called the blood-brain barrier. That's why water-soluble things, getting back to that idea of water versus fat-soluble, water-soluble antibiotics, water-soluble medications can't get to the brain directly. You have to either attach them to something that can carry through the membrane. The barrier. Very interesting. All of that becomes part of that. Something we touch on here in AP1. Okay. But sometimes they're always open, but most of them at signals or in the abundance of chemicals or certain conditions will either open to allow stuff in or close. Some are always open. Some leak. Some have little gateways. So you're going to get sick and tired of it. It's coming called leakage channels and gated channels. Channels that are controlled by electrical charge. They're called voltage-gated channels. They're controlled by chemicals. They're called ligand-gated channels. You're just going to hate me. Thankfully, that comes in the second 8 weeks when you can hate me. Speaking of that, some of you have lab iron. Are we going to be busy? So much. Okay. There goes your weekend. It's okay. See, when you're in students in one of these disciplines, it's like, eating, sleep, study, pick two. It's okay. It gets worse. But Dr. Robbins is wonderful, extraordinary man, extraordinary human being. And he's going to get it done. And you will learn a great deal. He totally revamped our laboratories, totally, on his own. It's just remarkable. It was extraordinarily disorganized, and he really took the reins and worked. So, just remarkably great job on that. Almost done with that. It's a stream of consciousness. Okay, so because of those really constitute boundaries, and just like our external, we have internal boundaries, they respond to stimuli. We talk about bacteria, simple organisms. They just are attracted to health. We respond differently. Sometimes that stimuli will provoke a specific response. Sometimes it will be more generalized. It's the kind of thing, depending if we have to respond quickly, it'll be part of our nervous system. If we have a little more time to respond. Sometimes it's our endocrine system. We get to aspects of that as well that are there. And so respond to stimuli or changes in the environment. Energy transformations, I call them. A lot of people just talk about metabolism. We have to have energy for everything we do. Metabolism is all of the body's reactions. They are broken into two pieces: anabolism and catabolism. Anabolism means we're making bonds, storing energy, we're building things up. Anabolism, think anabolic steroids, like bodybuilding steroids. Catabolism, think catastrophe, we break something down. And then we release energy. And we're doing it all the time. If you had time to grab breakfast this morning, maybe grab the protein. Effectively, some of that material is going to be an energy source. Some of it's going to be constructing, replacing materials that we have in our bodies. Others is going to provide the energy to do so. We balance that out. That's really what it is. I call it energy transformations, because I think it's easier for me to understand, hopefully for you to understand, be able to do that. And the energy transformations, as you'll see, involve a lot of different things. We have to bring it in, we have to break it down, we have to utilize the energy. And make no mistake, we are an extraordinarily efficient organism at utilizing energy, but it's not 100%. Our principal energy comes from carbohydrates. Indeed, most of what we ingest is carbohydrates, complex and simple. Even the proteins, when they're broken down, effectively enter a cycle as part of that carbohydrate utilization cycle. You've heard of it, probably. It's called the Krebs cycle. Thankfully, you won't have to know about it. But as much as it's an efficient cycle, for every unit of glucose, and basically all roads kind of lead to glucose, you'll hear me say, it only makes 40% efficiency. 60% of that energy is lost as heat. Even in a relatively energy efficient organism. If you're a big business, you'd be in trouble. I mean, if you're taking all this energy and you're only getting 40% of it being utilized for profit, and you're wasting the other 60%. Fortunately, it works for us. Just to give you an idea. And then lastly, you can see on all of these, you're going to see we have to have the capability for growth and development. And they're really important, both from passage from the fetus to us after birth. And then really the changes that we see in cells and things like that. That's just a construct that I wanted to give you in a sort of global introduction. I'll explain some of my other scribblings as we go along. We'll flip back to this a couple of times. What's nice about, and one of the reasons I guess that they picked this text is because it had clinical applications. Yeah, look. Yeah, I know it would shock you to think I'm not that far removed from being a student, but no, most teachers are not. We're always in classrooms. Just to understand, the context is important because a lot of you, why do I have to know this? Is this going to make me a better, whatever my career choice is? Am I going to be a better nurse because I understand this? No, not necessarily, but the context is important. No one's going to ask, they're never going to ask you, whatever your career path is in one of those things, about the Krebs cycle. They're just not. They're happy if you can help them, if you can take care of them, and you're a professional, and you're able to do that in a way, and you're user-friendly. Believe me. They're never going to ask you what you got on the NCLEX or a test like that. They don't care. Can you help me? Okay? But this gives you an idea of why we're studying this, because periodically they're going to ask you things, and if you can explain how something works, even in a very simplistic fashion, it really reinforces a patient-caregiver bond.
For all caregivers, doesn't matter. Doesn't be a doctor. Anybody's providing care. So all of those things are there for that. And understand there's nothing wrong with saying, I don't know. Don't ever try to tell, don't, if you don't know what you're talking about, I don't know, but I'll try to find out. It works really well. People appreciate that. Nevertheless, here, these are things, and this is sort of the basis, understanding how computer enhancement changed things. How we went from a non-computer age to a computer age, and it started. I mean, ultrasound's been around forever. Ultrasound is high frequency sound waves. It's been used therapeutically in physical therapy for more than 100 years. You generate those sound waves. They penetrate through the skin. They go through different kinds of tissues. When they get to thicker tissues, they reverberate. That reverberation basically generates heat. That heat, whenever you generate heat, it induces circulation to the area. That's how we used, for years we used whirlpool and ultrasound, hot packs and ultrasound to deal with a myriad of sports, industrial, different kinds of accidents and rehabilitation. They still do. But in the computer age, we took that ability to shoot in the sound waves, realizing it goes off of structures with different densities. So muscle is thick. It bounces off and reverberates it. Okay. What's in blood vessels, and that's why it's a good illustration, is fluid, blood. Blood is not uniform by any means, but by the same token, you're obviously going to get a different signal. We can analyze that signal. We can actually hear that signal. That's where the sound comes from. There was a device that came out when I was either just started it, got my doctorate, which was 1977, by the way. So don't look at me. Don't judge me. Okay. And it was like a little stethoscope that had an ultrasound device on the tip of it, and you put that on the blood vessel and you could actually hear. It's called the Doppler. It's used all the time nowadays in blood vessel when we're doing circulatory analysis for veins and arteries. You hear sounds. You'd hear a normal sound, and then you hear when the sound kind of had a different pitch, and that would tell you the blood vessel is narrowed. That would be nearing having a clot, potentially having something tragic happening to it. We use the same technique. But we took that technology and then added computers to it, and all of a sudden now we can visualize the fetus. Now we can see if someone has a valvular defect, if someone has a heart murmur, it will actually show it. They do a sonography, okay? They do what's called an echocardiogram. And the echo, think of it that way, is the reflection of that sound, except now it's just computer analyzed. See, that's how it works. It's not rocket science, not that complicated. It's understand that that's how those things work. And it's I mean, you're able to see things intrauterine. And we're not just talking about the gender reveal stuff, but you get a pretty good idea. And it's even facilitated having to do real advancements and having to even do procedures on the fetus early on. I mean, it used to be pretty hard to do those things. And most of the time this is what we call non-invasive. In the old days, they had, in order to find some things out, they had to do amniocentesis or various other, I'm trying to think of the other one that was utilized. And they were trying to find out whether there were abnormal cells or abnormalities where it could point to certain problems that were occurring with that particular fetus as it was growing in the intrauterine environment. Very interesting stuff. MRIs, magnetic resonance scans. Oh my goodness. I mean, brilliant. They're wonderful for soft tissue. You can see tumors, you can see fluids. Sports, every time any athlete has a problem, they're immediately going to go for a scan. You can rapidly identify. We didn't have that technology. We rapidly identify whether or not there's something like an ACL or a PCL tear. You should be learning about that. We're going to talk about it anyway here, because I do a pretty good size unit on that. That's the anterior and posterior cruciate ligaments. Cruciate means they crisscross, and you can see it. You can actually see the tear that's on there. We didn't have that. Fiber optics were in its early stages, and we were doing fiber optic procedures when I was a resident, and I was basically an orthopedic resident. So it's just fascinating. But what was before that MRI magically came about? The MRI was something that was used in a chemistry lab. Okay, it was a way we were able to identify organic compounds. It was used, it was called quantitative or qualitative organic analysis. You dissolve them in a fluid. The fluid was a special fluid because you're basically watching what resonance happens. You took the fluid, a tube of it with a chemical, you wanted to figure out what it was. Magnets spun around it, and you got a reading, a sheet of paper that was constantly moving. Okay, and what it was doing is the protons, the hydrogen atoms. So when we look at bonds without going to the chemistry, you have lots of C's and lots of H's. The H's, when the energy would cause the hydrogens to kind of move away, they'd absorb the energy. When they fell back down, when you remove the energy, they'd release energy. That's what resonance is. And so we would use that when I was a chemistry major to be able to see all that stuff. Little did we know that 10 years later, magically they were going to get a computer algorithm like they did with X-rays with something called CT, computerized tomography, because tomography was just X-rays at different angles. Computerizing that, using an algorithm, and magically we could see inside. And different levels, depending where we got it when it first started to spin or when the energy went out of it. So you get what we call type 1 and type 2 weighted images. It's fascinating stuff. And I assume they're still doing the nuclear. I know they're doing away with the quote nuclear medicine major, but I think they're still incorporating that in the health science sphere. And I've had some absolutely wonderful students from that over the years. My colleague, Dr. Buxton, and I teach physics. We were sort of put together a course in radiation biology that they took, and we were teaching it for a couple of semesters here. Nevertheless, fascinating. And it's about spinning. You're going to see some of that chemistry. No, you will not. This is not chem class in high school. We're not going to draw it to see you're happy. You're not going to have to draw a dot. We're not going to do stoichiometry. You're not going to know how many moles there are. Okay? Any of that stuff. You don't have to do it. I once tried to do that with students. They knew where my car was. It got ugly. I'm joking. They're all looking at me going, maybe he's not joking. It's everness. So we talked about that. All living things are made out of cells. Okay, internal environment. That's the thing. It's stable. That's when we get to homeostasis. When you take your temperature, when you take a patient's temperature, it is a snapshot. When you do a finger prick for glucose, it's a snapshot. We have to put context. Was it a fasting glucose? Was that temperature first thing in the morning? Was it temperature late at night? Had they just eaten? Had they not eaten? Is there something else going on and we just give them medication? If your temperature and mine varies during the day. I'm up and down, jumping around. Thank goodness I'm getting some activity. My wife, thank you. She often tells me to do things. Again, I know how to answer her. Yes, dear. Right away, dear. She can't talk to me like that. Take the garbage out. Yes, dear. Which is why I have survived to this advanced age. Everybody gives me orders. I tell you about my oldest. Well, he's a career officer in the Navy. All he does is give me orders. I'm on the ship. He's yelling at me. On naval vessels, they have to be. You have to seal any doorway you go through because it creates watertight. And I'm walking through. He goes, "Dog it!" That's what they meant by spinning the wheel and dogging it. She goes, "Look, mister," he's talking to his dad. He's yelling at me. Kids. Nevertheless. So all of those kinds of things are really important that are there. The maintenance of the internal environment. That's part of it. And so homeostasis, which we'll get to and where it is, cells are all interdependent. I was using the analogy in the earlier class about bone marrow. In bone marrow is where all of your blood cells, red cells, white cells, and there's a variety of each. White cells, there's red cells, there's platelets, are all produced from the same stem cell, from the same megacell that's there. That's sort of, but because of the lining, there's a particular kind of lining in the bone marrow that's draped over the interiors of the bones. If that's not there, and moreover, the structure of the interior of the bone marrow is tons of nooks and crannies, surface area. You'll be fascinated. I love it. The body is designed in such a way to maximize surface area in a little space. Your brain does it. It folds and folds and folds. You'll see it in the laboratory when you dissect a brain. We have billions, if not trillions, of cells folded over on one another because we have a relatively small container for that. You have 30 feet of bowel. Relatively small container. It goes from your mouth to your anus, and most of it's in here. It's all folded, twisted, folded, etc., like that. So it compacts into that area. It's fascinating. So there's a lot of when you go back to the bone marrow, it relies on those kinds of cells and the lining that's draped over them for the bone marrow to function properly. And bone marrow is very sensitive stuff. So things as you go farther in your careers, you'll learn about bad things like cancers. I brought that up when we get into the idea that they're going to talk about shortly, called differentiation. I understand for those who have lab, and I realize some of you are having it this afternoon and this evening. Okay. I'll be over in that room next door between 3 and 5:30, teaching another set of you folks for the first time. So if you want to break from Dr. Robbins, come over and wait. You're coming in there. Tell him I said hi. I always say hi to him. Is to understand that you talked about lining cells, and that's where we're going to get into what the distinction between carcinomas and sarcomas. So there's a lot that goes on, and the relationship of structure and function. Here, just to give you some idea, body is a very efficient mechanism. All of those, taking the energy doesn't just go to store energy and release it. The body is very selfish. It doesn't like to utilize its energy other than for essential functions. Most of what we do, most of that's moving things in and out of cells. The oxygen and the carbon dioxide. Oxygen going in, carbon dioxide going out, oxygen going out, and coming in the environment, carbon dioxide going out in your lungs. We rely on concentration gradients. Good old higher to lower concentration. That's what drives the bus. We're going to talk endlessly about movement across membranes, and movement from place to place is concentration gradient. And the beauty of this is we're very selfish in the sense that even if we have to spend energy, we try to make more out of it. So for instance, in the aforementioned Krebs cycle, another thing is we utilize something to generate all of this energy and store it in something called ATP, which we'll get to by way of something called the proton motive force. No, you are not going to have to know the Krebs cycle. Again, it's contextual information. I don't want anger in the classroom. It's not a problem. My colleague who was just teaching next door in the other room, that's her bailiwick to the measures. What happens is we take, basically what it is if this is a membrane, we generate protons from gobbling up mostly carbohydrates, okay, all those carbon and hydrogen bonds we're going to talk about, and we take those protons and we kick them over there from one side of the membrane to another, inside something called the mitochondria. So we have an abundance on one side and not very much on the other side. That's a gradient, from higher concentration to lower concentration. We have a specialized protein that acts kind of like a turbine, like we would go to Niagara Falls and generate energy. So as the protons now want to get back over from higher to lower to balance it out, they spin the turbine, and basically it creates energy. That's only part of it. We're spending it. Why are we pushing them up there? It creates another form of force. We have a lot of positives over here, very few over here. So relatively speaking, that's positive, this is negative. So it's like a double bang for your buck. We're moving it to create additional movement or additional force or generate additional energy. So the chemi-osmotic theory won a Nobel Prize. And we began to understand that. Just for instance, about importance. In your kidney, which you talk about in AP2, is very complicated. Basically what we do is we get rid of stuff we don't need. We actually reabsorb stuff early in the kidneys to spit it out later, because we can pull stuff up. We create a gradient, and we can pull more bad stuff out when we spit it out. We can do something called co-transport. And sometimes at the same time, it's almost like you can think of, and a swimming pool in a race, how swimmers will get in certain positions, because let the person in front until they're ready to pass them. Effectively, we utilize this. We can create those waves and those currents to co-transport and use them backwards to anti-transport. Very conservative use of energy. That's what the body can do. Are you going to have to know all of that? Are you going to have to understand a little bit about those things? Yes. So I'm trying to present it to you in that way so you understand these kinds of things. And I saw a few mistakes. Maybe you're the one I told you, don't rely on the PowerPoint. Did we have that discussion? Look, I'm user-friendly. If you want to come to me and say, you said this in class, but you marked it wrong on the test, okay. If I find that's true, I'm good. Don't come to somebody and say, the PowerPoint said this. PowerPoints are not proof positive. Textbooks, passages, scientific articles, they work because they're subject to what's called editorial review. They hire somebody to make the PowerPoints. I find tons of mistakes on PowerPoints. They summarize things, or they jump to conclusions which are not scientifically. Not just for me. I'll tell you about it, and I'll justify my approach to it by citing what's going on there. You'll get an idea of what instructors, what professors, who you can approach that way, and the ones who are approachable and the ones who will bite your head off over your. I know, and there are biters. In general, I like to go home and look. And I like to look at the person I look at the mirror. I like to be comfortable with. That's just me. So moving on. So that's this cell differentiation. That's the part you learned about. Some of you, or you will today. Lining cells, right? Simple cells. They are, relatively speaking, if we talk about a stem cell, like cord blood, or that cell I was talking about in the bone marrow, that really hasn't even begun to differentiate. So it's, unfortunately, it can be altered relatively easily. Lining cells that line your heart, that line your lung, that are on the skin, that are in your glands. Those kinds of cells, unfortunately, let me start with fortunately, they constantly replicate. Okay? Even your bone cells. I mean, your bones have been replaced ×2 already by your age. All the bones in your body. Okay? So these cells are constantly being replaced. You're shedding skin cells as we speak. Just do this. They're skin cells. You can't see them, but they're going. Cells in your mouth are constantly there. That's the beauty of cellular differentiation. If you have lining cells, they're easy to replace. If you scrape your skin just a little bit, it's going to heal. Nobody's going to know where that was. If you're young, it'll go right away. Probably would have healed by now. Maybe it'll leave a little discoloration. And if I look next month, I probably won't remember where it was. Okay, aging does play a role in that. That's the good news. Cells that are just, relatively speaking, undifferentiated, like lining cells, and we're covered in lining cells. Every surface internally and externally has lining cells. The bad news is they replicate so rapidly, they changes in mitosis, damaging to that mechanism in the cell cycle, which we recover, results in cancer. The most common cancers are cancers of lining cells. Their last name is carcinoma. The most common system that's affected over a lifetime is your skin, mostly because of sun exposure. Thankfully, the most common skin cancer that we get to, it's called basal cell carcinoma. It's treated relatively simply by excision. It doesn't require chemo or radiation or additional care. So it's more cosmetic than anything else, thankfully. Not all are created equal. On the other hand, when you talk about connective tissues, which you may or may not have done, or maybe you touched on, we talked about collagen. Those are a lot more severe and dangerous. They're called sarcomas. So the difference between a glandular cancer, let's say, or an epithelial cell cancer, it's a carcinoma, versus a sarcoma. Now you have tissue that's much more highly organized. When something goes wrong there, it's just a bigger problem. Very significant. The most common cancers that we face, you and I, breast cancer and gland cancer, other than skin. Those are glandular cancers you'll see the name adeno. ADENO means gland. So they're adenocarcinomas. And at an alarming rate. I mean, unfortunately, we'll get to it, but the rates are scary. They're there. More frequently than 1 out of 10. 6%. Thankfully, that prostate cancer is one I have avoided. See, I'm sorry, you get stuck with lose health. It's a good teaching experiment. Don't smoke. Not that I'm telling you something. I smoked for 40 years, 2 packs a day. I have not had a cigarette in the last 17 years and 2 months plus. 2 months and 7 days. Well, I know that is worthy of it. I can pat myself on the back for that. Thank my wife. That's why I'm still here. But I got a reward for that, not lung cancer, bladder cancer. Back about 10 years ago. I'm doing good now. That was no picnic treating. Scared the heck out of my kids. Scared the heck out of me. Moving on. So, differentiation. For instance, the good news about cellular differentiation is this. Heart muscle is highly differentiated. It can't replicate anymore. The problem with that, if it's damaged, it can't replace itself. All it can do is try to repair with scar tissue. We have a word for that. Most of the time, heart muscle is damaged. It's called an infarction. Death of tissue from lack of blood supply. Infection has to be with the microorganisms. Okay, infarction is something that tissue is so fragile. If it loses its oxygen for a significant period of time, it doesn't have to be long, it dies. And unlike lining tissues, your skin heals with scar tissue. It can be silent, it can be deadly, it can be something that can be treated or not, but it will always be damaged. No, I mean, oh, mom, he had a very light heart attack. Nothing's wrong. Some of the heart muscle died. Whether or not they have been significantly affected, that's a whole other discussion. A lot of people find out later in life when they have a scan or something done for maybe an insurance purpose or because of a check they're having with their physician. There's evidence you had a heart attack back X number of years ago. I didn't know that. But the reality is, yes, it's gonna happen. So that's part of that differentiation. We'll talk endlessly about movement across membranes. Okay, a lot of it has to do with, and let me just do the communication while I'm thinking about it. The cell-to-cell communication, that heart muscle. Love the fact that they had that. All those cells communicate right there. They send signals when one part contracts, the next part contracts, and the next part contracts sequentially. That's why those defibrillators work. We fire them all at once, all the heart muscles relax, hopefully the pacemaker is intact, and it will resume its beating. That's the whole idea of defibrillation. Certain rhythms are, and you learn a little bit about that in AP2 and AP2 lab. Okay, the contact, because those cells are constantly moving, they have to be told when to move, and so they don't peel apart. That's a very special contact you learn about in AP2. It's called an intercalated disc. It's like riveted together because you're constantly moving. So it's really neat stuff when you see that. I'm trying to get across to you in the way I do these presentations. I know it's a little nutty and I get off the track. I'm trying, my teaching philosophy is very simple. If I'm not enthusiastic, why would you be? If I'm not showing you that I'm interested, I could read this stuff to you. You're going to have, unfortunately, we all have it. Cell membrane mechanisms determine entry of substances and respond to signals. Yeah, we can do that. We don't want to do that. What's neat about understanding things, when you talk about cells in the environment, we're going to get down the road to a concept called tonicity. And we're not talking about like muscle tone. The tonicity in the context of circulation has to do with the amount of things dissolved in water inside body fluids inside the cell. Even though the fluids are different than what's inside of them, they're the same in the amount of stuff that's dissolved based on how much water there is. It's a proportion. It's a simple ratio. And that is a number that stays the same. So, for instance, if you work out and you're outside, you're a little dehydrated, you rehydrate to restore that number. Okay? If you're overhydrated, to restore that number will compensate. So you're going to hear about, and this is why you're so careful in the hospital that we use things that are same, isotonic or have isotonicity. The most famous of those fluids that we use when we want to give medication is called normal saline. We'll learn about what makes it normal. What makes it normal, effectively, the short course, it has the same tonicity, the same relationship of stuff dissolved in the water to the amount of water. Fine, that's what we use. Sometimes we use variations of that. But that way we're relatively sure, like say in an emergency room situation, that we're not going to, until we know more, we're not going to push too much stuff in or pull too much stuff out. Because it'll either suck stuff out of the cells and they'll shrivel, or it'll push fluid into the cells and they'll burst. Not a good thing. Okay. So that's part of it. So determining the entrance, yes, signals are all part of that. We'll get to the feedback loops for homeostasis. They're constant. We're going to talk about negative feedbacks that make subtle adjustments and take it back to what we call the set point, which we'll get to next time, and ones that don't. Well, positive feedback. Thankfully, it's an easy question to answer if you forget. Nearly all of them are negative feedback. I'm fond of telling students, remember exceptions. If the vast majority of things are negative feedback and you don't remember what one is that they ask you about, let's say they're asking about glucose metabolism. Well, I don't know. The default is negative. Sometimes it's easy to do that. When you have a lot of stuff to study, when you're doing the bones in lab, which bones are not part of this might be just as easy as which bones are part of this. So a lot of times and that's the way you memorize stuff. You put it in packages. Body work, that rote memorization when you're going femur, head of femur, head of femur, head of femur, neck of femur, greater trochanter, lesser trochanter. And you keep repeating yourself, it doesn't stick with you. Very, very briefly. That cramming, no bueno. A word to the wise. This is not a course where the night before you're going to be able to study for this test. Forget it. Don't do it. You just blow it. So fine, you're not in Kansas anymore. The tests are a lot harder. They are, everybody, not just mine. So if you were, in high school, I could just, look at my notes the night before, fine, watch it on TV, same time. Don't work. Don't fall into the trap. You'll see people who fall into the trap. You won't see them for long. Don't be one. So we know about replacement, elimination, energy, blah, blah, blah. Growth and reproduction. We touched on reproduction. Yes. We not only need new of us to keep the species going, but we mostly need new cells. A lot of growth and development, it's like that differentiation, they're part of it. Growth in size, differentiation, growth in number, changes between the fetal environment and after a baby's born. It's very, very interesting. Sometimes things stick around longer. You'd be surprised as we look at more of it, but that's an important part, and certainly responding to stimuli. I mean, organisms, again, like bacteria, it's either attracted or repelled by. They have no decision. They have no eyesight. Anything along those lines. You and I can decide to respond in different ways. I mean, if it's warm, we can just change our clothes or remove layers of clothing. Or by the same token, we can flip the AC on, turn the heat off. I mean, there's lots of things that we can respond in ways that we want. And certainly movement is somewhat self-explanatory. I don't go crazy about this because we touched on it. This is where there's some mistakes. Respiration is not we don't make energy. Rule number one it's either laws of physics or laws of chemistry. I think it's energy can neither be created or destroyed, only transformed. The energy is there. Our energy comes in the stuff that we ingest. You eat or drink. Period. End of sentence. We transform it. Most of it we transform, most of it we lose is heat, a form of energy. Okay, and every time you do an energy transformation, we lose it. That whole entropy thing, you know, I forget which name it is. I was a chemistry major, this is ages ago. I got my chemistry degree 52 years ago. It's a while, I know. Be happy I remember the electrons and the protons. So it can be, so that's stupid. Now, if you're going to tell me that part of the ability for energy transformations, respiration is twofold: pulmonary, exhaling a waste product, inhaling oxygen, which is necessary to do energy transformations, I'll buy it. Or at the cellular respiration, it's similar. We're depositing that oxygen, it's going into the cells so we can do the energy transformation, and we're absorbing some of the waste product to get rid of. We're absorbing most of the waste product, carbon dioxide, and a certain amount of water, which are the waste products. Okay. Digestion and absorption are not interchangeable terms. Digestion is both mechanical and chemical, and it's how we break down substances that we're going to utilize into the smallest usable component. We can't, that's what lactose intolerance is about. Lactose is a sugar that we see with dairy products. When we're born, frequently do not have that enzyme, and some people don't develop that enzyme ever, and some develop it at very different times, or it goes away. And because of that, we have to break that up into smaller components. Lactose has two sugars bonded together, which we'll learn about, but we can't utilize it, and if we can't utilize it, it creates havoc in the digestion. And we'll get into what lactose intolerance is. So that's part of digestion. Absorption is that final product. You'll see when we absorb something called amino acids. Now, so when you have a protein bar, it's not the protein. We're breaking it up into amino acids that we utilize that. And then excretion is pretty straightforward. Yeah, water, we're big bags of water, blah, 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 blah. Food, straightforward. Oxygen, I don't know if it releases energy from food. Again, it's an essential part of energy transformations. We have to have oxygen. We have to have water to break. We do everything by hydrolysis to generate and release energy, and we have to have oxygen, which has a different role in the mitochondria. It has to do with electrons. So you're not going to have to know about it, but you're going to have to know a little bit about it in that regard. It's called the terminal electron acceptor. And this is the idea of those fluids. And they're not the same, even though they're similar based on how much stuff is dissolved. For instance, inside your cells, you have more potassium. Inside your bloodstream and inside what we call the interstitial or extracellular fluid. Interstitial means in between the tissues. You have more sodium comparatively to the amount of potassium. More sodium outside, more potassium inside. And the reverse is true. And this is pretty straightforward. We'll get to that with pressure, and then we end it here with homeostatic mechanisms. Have a great day. I will see you all on Friday. Like I said, if you want to come and wave, you're coming either leaving lab or going into lab, I'll be over there at 264. It's all right. You can ignore me. I'm good with that.